The main pattern of motility that we have in the small intestine is segmental contractions, also known as segmentation. Okay, so the main outcome of segmentation is mixing. So the purpose of segmentation is to mix, and you do get a little bit of propulsion from that as well, a little bit of forward movement of the contents of the small intestine. This figure here shows us how segmentation works. Basically, the way it works is you contract some of your circular muscles in your intestinal wall, okay? Then you contract other muscles, right? So you kind of take turns contracting one set of muscles and then another set of muscles. And in that way, you end up mixing up the contents of the small intestine. You can kind of liken that to, so remember when I talked about peristalsis, I talked about how that was like pushing up from the bottom of a tube of toothpaste, right? So I have a tube of toothpaste here to demonstrate, right? So peristalsis is like this. It's like pushing up from the bottom to move the toothpaste forward, right? Segmentation is different. Basically what you're doing is you're kind of squeezing, at these points first, and then you are squeezing at these points here, and then you're switching back and forth like that, okay? And the result of that is mixing, okay? And if you have a 3-color toothpaste, such as Aquafresh, you can end up mixing up your 3 colors in that way, okay? Oh, by the way, this is toothpaste from Japan. It's called breath communication etiquette, or in Japanese here it says echiketto, okay? So just a little having fun here. Okay. So, but let's talk about this. So why? Why would you want to mix? Why is that useful? Why is it useful to mix up the food, the contents of the small intestine? Think about that for a second. Okay, so I've already addressed this before when talking about the stomach, so you hopefully already know this, but mixing is useful because it helps the contents of the chyme come into contact with the digestive enzymes. And in that way, the enzymes can work on the contents of the chyme to break them down and get them ready to be absorbed by the walls of the intestine. Okay, so now that I have introduced you to some of the basic processes that happen in the small intestine, we are ready to look at specific molecule types and see how they get processed by the small intestine. So let's do that, starting with carbohydrates, okay? So I would like to start by pointing out that we ingest carbohydrates in many forms. We ingest them as polysaccharides, as disaccharides, as monosaccharides. And what we need to do is we need to break those down into monosaccharides because that is the form that we are capable of absorbing into our bodies, okay? So that process is shown here in this slide. Okay, so we have our carbohydrates in our different forms, polysaccharides, disaccharides, monosaccharides. We have a variety of enzymes that break those down into the component monosaccharides, okay? I'm not going to hold you responsible for knowing the specific names, but basically, yeah, as long as you know that we're breaking down into individual monosaccharides, that is what I want you to know. And then once we break those down into monosaccharides, we transport them across the epithelium of our small intestine. So that's shown here. This is our intestinal mucosa. We use a variety of transport mechanisms. Again, I'm not going to ask you to know the details of that, but you can see these examples of different ways that we are transporting our monosaccharides across that epithelial cell layer. And so I just want to remind you that in order to move a molecule across the epithelial cell, we actually have 2 crossings that we need to do, right? So we first need to cross the apical membrane here, and then we cross the basolateral membrane. That brings our monosaccharide to the interstitial space where it then can be absorbed into the bloodstream. Okay. All right, so that was our carbohydrates. Now let's take a look at how we digest and absorb proteins. Um, our proteins are broken down into individual parts, so single individual amino acids or very short peptides like di and tripeptides, and occasionally slightly longer peptides as well. Okay, they're broken down by by enzymes called peptidases, and we have two main types of peptidases. We have what are called endopeptidases, which will just kind of cleave a polypeptide chain in the middle of the chain, right? So here's an example. We have this chain here. We are cutting right in the middle there, and then we end up with two shorter chains. And in addition to that, we have what are called exopeptidases, which basically work by taking off one amino acid at a time. Okay, so here we have a polypeptide chain here. We have a type of exopeptidase called a carboxypeptidase, which is going to take off the amino acid from the carboxy terminal, right? So that's what's happening here. We're chopping off this amino acid to give us that amino acid, but freed from the rest of the peptide. And then we have aminopeptidases, which are going to chop off individual amino acids from the N terminus, right? So we have our amino acid being freed from the rest of the peptide over here. Okay, so we have individual amino acid, and the rest of the peptide here. Okay. I do want to remind you that some of this process does start in the stomach. Remember that we had pepsin that was going to start to break down some of the proteins, but we also have a lot of this happening in the small intestine as well. Okay, so once we have broken down our proteins into smaller components, either individual amino acids or di or tripeptides or occasionally longer peptides, we are then ready to absorb them. Okay, our proteins are mostly absorbed in the form of free amino acids. Okay, so that is shown here. This is kind of a busy figure, but that's what's shown here is an individual amino acid being transported across the wall of the small intestine. Sometimes we absorb in the form of di and tripeptides. So those are shown here. Okay, again, I'm not asking you to know the specific transport mechanisms. Okay, and then very rarely we can also absorb longer peptides by transcytosis. So that's what's shown here. We have this longer peptide, it's being endocytosed at the apical membrane. Again, this is the lumen of the small intestine up here. We're endocytosing at the apical membrane, transporting it across that epithelial cell layer, and then exocytosing at the basolateral membrane, where it then can enter the bloodstream. So that's where all of these little protein byproducts are entering the bloodstream.